Now we move into 2.5D. So this is like the level of already like tertiary interactions, but people tend to still represent in 2D, not in 3D. And there are two main components of 2.5D. First of all is non-canonical base pairs, so all the base pairs that are not Watson-Crick or Wobble-GU base pairs. And the second is coaxial stacking, so we will first start with non-canonical base pairs. So apart from three canonical base pair types that we attribute to secondary structure, this is AU or AT in DNA, AU, Watson-Crick AU and GC and Wobble-GU. We also have multiple non-canonical types of base pairs. So where we have still two bases located in a single plane, forming hydrogen bonds with each other, but they are like oriented differently. So examples here are like Hooksian base pairs. They are still AU, like these examples are still AU and GC base pairs, but here the purines interact with the pyrimidians with their Hooksian edge instead of Watson-Crick edge. So like they are like rotated with respect to the Watson-Crick base pairs. And here are the examples of homopurine pairs where two purines, AG or AA, interact with each other. So we have all the variety of base pairs, like different orientations and different base types. We have purine-purine base pairs, pyrimidine-pyrimidine base pairs, and various versions of pyrimidine-purine base pairs as well. One of the first classifications of all these non-canonical base pairs came from Sanger classification from 1984. It included 28 types of different base pairs that include at least two hydrogen bonds. So this is the figure, with Rome numbers. We have the 28 types. Watson-Crick base pairs are 19 and 20. So this is GC 19 and AU 20, and the Wobble-GU is number 28. Later in 21st century, Xanjan Liu, the developer of DSSR, actually discovered, thanks to DSSR, that Sanger missed one more type. So this is like a CG base pair with two hydrogen bonds that is missing from the Sanger classification. So Xanjan Liu annotated it as type 29. And maybe we also missed some more types as well. So you can try to find more if we're missing anything. But this classification was just basically cataloging or numbering the base pairs. It was not very useful. So it was useful for the identification of the base pairs, but not for their classes. And then in the beginning of 21st century, Leontis and Westhoff introduced their classification, which is Leontis-Westhoff classification, which is geometry based. So for each residue, for each nucleotide, they distinguish three interacting edges. So Watson-Crick edge, which forms the Watson-Crick base pairs, Hooksian edge, which forms Hooksian base pairs, and sugar edge, because it includes ribose. Ribose is a sugar, and in sugar edge, sugar edge is the edge of a base, but it also usually includes auto prime atom of a ribose. That's why it's named sugar. So we have three interacting edges, H, W, S. And we have two possible orientation of glycosidic bonds. So if they're co-oriented in the same way, then it's cis base pair. If they are oriented in the opposite way from the base pair plane, from the base pair axis, then we call it a trans base pair. So, and this cis or trans and two interacting edges give us a geometric classification of base pair families. So these are the families. So here we have 1, 2, 3, 4, 5, 6, 12 possible families with respect to symmetry. So we have only one TWW and only one TWH here, because HW is included here. Once we, until we exclude, as long as we exclude the base types, we actually have 12 geometric families. And once we consider symmetry, we have 18 possible relationships. Here we have notation for diagrams for these base pairs. So we have field shapes for cis base pairs, empty shapes for trans base pairs, Watson-Crick edges depicted with circles, Hooksian edges depicted with squares, and sugar edges depicted with triangles. And of course, once we consider base types, we have like more than 100 of different base pair classes. So this is just an example of a representation. This is a base triple of 2 base pairs. So here we have Hookstein, Watson, trans Hookstein, Watson Crick, AU base pair. Here we have cis Hookstein sugar, UG base pair. Note that like Hookstein sugar UG differs from Hookstein sugar GU. So here the order of the bases is important because it differentiates which edge of the base interacts. And now this base pair classification is more or less like widely accepted. So in many papers we have the diagrams of secondary structure with tertiary base pairs represented in Watson Crick in Leonsis-Westhoff representation. So this is an example of E-loop motif. This is Sartzen-Riesen loop. So here we have characteristic Hookstein sugar AG base pairs, or Hookstein AA base pair here. And these diagrams help us actually to compare the structures with each other, and this is exactly like the reason why it's attributed to 2.5D. So it's still like a secondary structure diagram, but it includes tertiary base pairs. Or this is like a diagram of this king-turn motif. Also we have like this is how we depict canonical base pairs of CWW type. So the Watson Crick base pairs in this classification are cis Watson Crick, so CVV. And their field shapes, and these are 4 non-canonical base pairs in this diagram. So this is very, very useful. And another feature of this classification is that geometric families kind of hint us the interchangeability of the base pairs. So like if in some position we have a CVV, and in homologous structure we also have a CVV, but with different base types, we still can can hypothesize that like the geometry of these base pairs is similar. And this the concept of this interchangeability is called isostericity. It was coined by, I believe, by Westhoff. So the concept of isostericity says that like 2 base pairs are isosteric if, when we superimpose them, the C1' atoms of riboses match perfectly. So, and the thing is that in Watson Crick GC and AU base pairs, these 2 types are perfectly isosteric with the distance between C1' atoms like a fixed value of like 10.5, 10.6 angstrom in both types. So this is why these 2 Watson Crick base pairs can form long stacked helices. And actually wobble GU base pair is not perfectly isosteric, but very like closely isosteric to Watson Crick base pairs with a C1' distance of slightly more, like I believe 11 angstroms. And this is an example of non-canonical base pairs that are isosteric, and this is an example of 2 non-canonical base pairs that are not isosteric. So once we superimpose them, the C1' atoms do not match. And so this classification of geometric families promoted the studies of isostericity, and now we can actually from real structures, comparing the families of different base pairs, we can analyze what geometric families are isosteric depending on the base types, and which are not. So there is a whole computational problem of base pair annotation. So all these non-canonical base pairs, once we have a 3D structure, experimentally determined 3D structure of RNA, we want to derive like what non-canonical base pairs are there in this structure. And it's not a trivial task, because the bases of a base pair are not perfectly normally are not perfectly located in a single plane with a canonical like standard geometry. And for that reason, we have a task of annotation, like how do we recognize that these bases do form a base pair or do not. And for this, different approaches use like different sets of parameters, different strategies. So commonly, like there are 2 main strategies. First is to first parameterize the base pair, like the given base doublet. So we call a base doublet is like each pair of bases which are not necessarily form a base pair, but are like close to each other, like which are base pair candidate. Right? So for each doublet, we can calculate like, for example, these 6 parameters, like 3 distances, shear, stretch, stagger, or 3 angles of how the bases of the planes of the bases are rotated relative to each other. And we have like a standard set of known base pairs for which we know the perfect parameters, and by judging the parameter values, we either annotate this doublet as a base pair or we do not. And the second strategy is when we have reference canonical base pairs and we just superimpose the reference base pairs to the candidate base pairs, and judging on the similarity, on the RMSD value, or like distance RMSD value or something else. If the candidate base pair is close enough to the reference, then we call it a base pair. And we have 5 commonly used base pair annotation tools. I will list of all 5 with the like format of their annotations in the order of their papers. So the first one was MC Annotate by François Major lab from 2001. This is like a snippet of the annotation. So we have a pair of bases like chain A residue 2, chain B residue 63. It's a GC base pair. It's like Watson-Crick base pairing. It's anti-parallel cis, and this is a Sanger classification number 19, 20 for AU base pairs, and so on. Next was RNA View by Eric Westhoff lab from 2003. This is again a snippet of the annotation. So it also annotates not only base pairs but stacks. So here we have like a stack between residue 5 and 6 of chain X. It's 2 pyrimidines. So here we also have for base pairs like cis or trans annotation, Sanger class. Here we have like edges for non-canonical base pairs. So for class 19 it's always plus plus, for class 20 it's always minus minus. We have some additional annotations of like base pair conformation. So here one of the residues is in cysteine conformation. So this was the second tool. Third tool is Fred from BJSU, from originally Nicholas Leontis lab and later Craig Zierber's lab. Fred is from 2008. The original Fred was implemented in MATLAB. Now the like fresh version of Fred implemented by Craig Zierber is Fred Python, implemented in Python. Also like pretty widely used tool. It annotates not only base pairs again, but base pairs, stackings, some base phosphate interactions, and some base ribose interactions, I believe, and some more. So this is a snippet. Here are the base pairs, CWW, CWW. These are base phosphate, so BPH is a base phosphate, also like classified into several classes. This is class 0. This S35 or S53 are the stackings. So this like 5 or 3 are the the orientation of the faces of the bases, whether they look at the 5' end or at the 3' end. And to the before the class of an interaction is stands for near. So these are like instances that are not perfectly close to the canonical geometries, but so do not pass all the thresholds, but kind of like pass the relaxed thresholds. So that's why they're like near this class of interaction. Now, later was introduced in 2014, introduced tool Klarna from Janusz Bunitsky lab. It also introduces many types of interactions, like these are stackings, base ribose, base phosphate, some diagonal classes, sandwich classes, or some like look at the paper for some additional classes of interactions. These are the base pairs of WW-cis. And the feature of this tool is that it's not a binary classification, but each instance has a score. So the higher the score, the more confident the tool is in the annotation. So this is like score 002 is pretty low, as you may understand, and the score of 0.9 is high. And later, Alexander Liu introduced tool JSSR, which is like a successor of X3DNA toolkit, which was originally designed for the annotation of DNA base pairs. So then DSSR came to life much later. This is again the snippet of base pairs. So we have like two residues, G and C, GC, like name of the base pair for some named base pairs. So we have, for example, a sheared, a Hookstein base pair, a Watson-Crick, a Wobble base pair, some more. Zenger class, Leontief-Westhoff class, and some extended Leontief-Westhoff class with the minus for anti-parallel orientations and plus for parallel orientations. Some values of the parameters, hydrogen bonds included in the base pairs, and so on. And this is the extended output of DSSR that I'm normally using. I normally use. So these are the 5 base pair annotation tools. Some of them include their own databases, like with the already performed annotations for all the known experimentally derived experimental structures. Experimentally determined RNA structures. So this is the database of DSSR annotations. This is RNA 3D Hub from BGSU, which is a database of not only but also like Fred annotations. So this is like Fred base pairs, and here we could look at some other types. Here we have loops again because it's also it has a motif atlas, a structure atlas, and some more tools as well. So it's truly a hub. Now regarding visualizations, so we have so-called circular diagrams, which is not frequently used type of visualization, but it's from a very recent paper from Craig Zirble, Red Seed. It's a part of RNA 3D Hub, and it has a circular diagram, and you can select like what types of non-canonical base pairs you want to look at, and you can click on the specific base pair and it will be shown in 3D with some neighbors and possible nucleotide numbers and so on. More often used representation is when non-canonical base pairs are embedded in secondary structure diagrams. So this is actually an example of a prototype of a visualization developed by Anton Petrov, the developer of R2DT, from the recent hackathon from Prague, organized by Bogdan Schneider. So this visualization includes like a 3D window of a 3D structure. It's R2DT powered 2D diagram with non-canonical base pairs. You can again filter by base pair types. You can click and see like the visualization in 3D, and it also has a dot bracket section where also like a clickable one when you can see all the base pairs of a given pair of residues. So once you click on this base pair, it will show like all the other pairings of the involved bases and so on. So I believe this prototype will soon be incorporated in many RNA related resources. So basically like the tertiary base pairs, non-canonical base pairs, all the base pairs that are not a part of secondary structure, we often represent them as part of secondary structure on secondary structure diagrams, depicting like the types. So here you can see again the types of non-canonical base pairs. And the second part, which is less named 2.5D, less attributed to 2.5D, but it should be, because it's also like an interaction that we normally depict on 2D secondary structure diagrams, but it's actually a part of 3D structure. So it's just like for non-canonical base pairs, and this is a concept of coaxial stacking. So as I've already mentioned, like a stack of consecutive base pairs, consecutive stacked base pairs is called a stem. And so in a single stem, all the base pairs are stacked as a stack of coins with each other. But sometimes non-consecutive base pairs also stack to each other. Like for example, like here we have 2 stems and 1 nucleotide. And in 3D, it would look like this nucleotide will be looked out somewhere here, and these 2 base pairs will also stack to each other, despite being non-consecutive in sequence. This is an example of a transfer RNA 3D structure, and this is its secondary structure. And you can see that like the 2 stems, like red and blue, stack on top of each other here and form like a continuous helix-like structure, like a continuous helix, but it's not a consecutive helix. And also we have a second coaxial stacking here, when a green and purple stem also stack on top of each other. So this is the coaxial stacking of 2 non-consecutive base pairs. It can be it can happen in different ways. So here is an example of four-way junction, and we, for example, can pair stems in different order. So here, light blue is paired with pink, and dark blue is paired with red. And here we have red with light blue and dark blue with pink. So the order in a single junction, in a single multiple junction, the order of coaxial stacking may be different. And so actually these diagrams show why I attribute coaxial stacking to 2.5D, because it's a secondary structure diagram which actually shows the coaxial stackings, right? And depending on the order of which stems stack with each other with a helix, we can distinguish different junction topologies. So this is a classification of three-way junction topologies, where we have a loop confined by three stems, and two of them may be stacked with each other, but it may be different too, right? And the third one can be perpendicular, can be parallel to one of them, can be parallel to a third one, to another. And this is an example of 3D structures of these different stackings. So here, helix 2 and helix 3 are stacked with each other, and H1 is not coaxially stacked. Here, instead, H1 and H2 stacked, and H3 is alone. Here again, H1 and H2 stacked, and H3 is it's in a different orientation. And the same for four-way junctions. So for four-way junctions, we have even more types. So the common ones, I think, this H and this X orientation. So it's in this classification, X is something else. So this is H, and everyone understands why this is H. And I tend to call this orientation X because we actually have an X here. And these are the two common junction topologies. So these are the two non-canonical base pairs, and coaxial stacking are the two types of concepts of RNA structure that are kind of in between of 2D and 3D. So now we move to 3D. So we already know that we have base pairing and base stacking interactions. And these are actually the two main drivers of RNA folding. So we have base stacking, where which is a hydrophobic interaction. So two planes of two bases stack with each other to forbid water to go there. So that's why it's hydrophobic. It's very stable. It's very strong interaction. And actually, base stacking is stronger than base pairing, I would say. So stacking is more important than base pairing. Base pairing is almost as important as base stacking. Base pairing is driven by hydrogen bonds. So here we have coplanar orientation in a single plane. Here we have coplanar orientation in neighboring planes. And apart from stacking of base pairs, single bases can also stack with each other. So we can have base pairs that are stacked with other base pairs. We can have bases that are not base paired but stacked with something else. And we can have base pairs which are not stacked with anything else. But of course, the most favorable orientation is a base pair that is stacked with something else. And on top of that, we actually have a variety of tertiary interactions that are neither stacking nor base pair. And I will cover that in the next slide. But first, so the main element of the 3D structure is still a helix, which is a stack of consecutive canonical base pairs. Important concepts here are that thanks to isostericity of canonical base pairs, Watson-Crick base pairs, and to some extent wobble base pair, so here actually the wobble GU base pair, thanks to isostericity of these types, they can form long helical fragments, unlike the non-canonical base pairs. And these helical fragments are always, or almost always, anti-parallel. So if one strand goes 5' to 3' in this direction, then the other strand goes in the opposite direction. And these helical fragments have two, we distinguish two sides of these helical fragments, which we call grooves. So if this is, I believe, DNA helix of B form, so we have a major groove and the minor groove. And this is actually the naming that comes from the B form DNA. So minor groove is smaller, that's why it's minor, and major groove is larger, that's why it's major. In RNA A form helical geometry, it's the opposite. So here we have a minor groove larger than the major groove. Easily to distinguish between the two grooves on the 3D visualization is to look at, you can by looking at auto prime oxygens. So the oxygens of the ribose, this one and this one, look in the minor groove direction, and the major groove does not have any ribose oxygens. And when you look at the base pair in this orientation, major groove is on the Hooksian edge side and minor groove is on the sugar edge side. So minor groove is where we have ribose oxygens. That's why the sugar edge corresponds to minor groove. That's an important option. Yeah. So now all the interactions formed on top of secondary, not on top of secondary structure, but on top of base stacking and base pairing interactions. And we have a variety of those, which are still frequent, but maybe a bit weaker than base stacking and base pairing. But we have a whole bunch of functional instances of these tertiary interactions, so they should not be ignored. So this is not the perfect figure of base ribose interaction. So here actually a base of this residue interacts with this oxygen of ribose of another residue. This is a classification of base phosphate interactions where we have an oxygen of a phosphate interacting, forming hydrogen bonds. Yeah, so here we have also hydrogen bonds of base and phosphate, here are hydrogen bonds of base and ribose. And these are two non-standard stacking interactions. So here we have so-called atom-base capping, which is a stacking of a base plane with a single atom. So most often it's O4 prime, but sometimes it's maybe phosphate oxygen as well. So it's capped, so the atom is capped on top of the base plane. And this is what is called like T-stacking. So which is basically an example of atom-base capping, but in this example, an atom is an atom of another base, right? So here, if the capping atom belongs to ribose or phosphate, we just call it atom-base capping. If it's an atom of another base, then it's called a T-stacking. And it should be obvious why it's called like that. So this orientation of two bases form a T shape. Now, and these interactions form like larger building blocks of RNA structures, which are called 3D motifs. So 3D motifs are like recurrent networks of interactions that can be found in different structural contexts. I will just list the most widespread and the most well-described examples. So this is an example of a GNRA tetraloop. This is a loop of 4 residues, so this is a hairpin loop in secondary structure terms. So this is a canonical base pair that confines, so usually it's a flanking base pair of a stem, the last base pair of a stem, and these are 4 residues that are continuous in the backbone. So the entire motif includes 6 residues because we usually include the canonical base pair here, but the loop itself has 4 residues, and the GNRA is the IUPAC pattern of the residues. So the first is always G, second can be any second nucleotide of the loop or third nucleotide of the motif. Can be any nucleotide. This is normally a purine R, and this is most often adenine, so it's A. So this is why it's GNRA, and this is an instance of GAAA, which is an example of GNRA pattern. And the characteristic interactions of this motif are the canonical base pair at the bottom, non-canonical base pair, which is of GA sugar Hooksian, so which is a trans sugar Hooksian, so TSH type of GA base pair in the stacked on top of the canonical base pair. This base pair also includes a base phosphate hydrogen bond of this guanosine. Then we have interaction of this sugar with this adenosine, with this base. So this is base ribose hydrogen bond. Then we have atom-base capping of a phosphate oxygen, this one. So this is the atom-base capping, this is base ribose hydrogen bond. And yeah, and this is actually the same thing, just oriented differently. Sorry. Yeah. And in turn, so all these motifs is a complete zoo. So we have larger motifs, we have smaller motifs, and some smaller motifs are like sub-motifs of larger motifs. So it's really like a whole modular hierarchy. And this just like as another example. So this generate tetraloop actually includes as a sub-motif a variant of a U-turn, and U-turn is a 3-nucleotide motif which forms like a sharp turn in the backbone. And most often it's of pattern U and R, so the first is uracil, then any nucleotide, then purine. It has this like ribose base characteristic hydrogen bond. And generate is a special like type of a U-turn where we have instead of uracil, the most common type, we have a guanosine, right? So the geometry of like U-turns and generates are like the resemblance here is uncanny. So we so we sometimes say that generate tetraloops include a U-turn as a sub-motif, right? So that's like how it all works. Now another, I think this is probably the most frequent and most well described tertiary motif in RNA is A minor. A minor is a base triple where we have a canonical, most often a canonical base pair. Here is an example of GC. And on its minor group side, so this is a reverted figure where we have ribose oxygens on top. With its minor group side, we have adenosine forming hydrogen bonds with its minor group side. That's why the naming A minor, so we have A interacting with the minor group of canonical base pair. We have two main types of A minor, so this is type 1, this is type 2, and they were introduced when they were identified in the first, in one of the first ribosomal RNA structures determined experimentally near the year 2000 or 2001. I don't remember from which lab, unfortunately. So yeah, so the two most frequent types are type 1 and type 2. In type 1, adenosine interacts with both residues, and in type 2, it interacts only with one residue of the base pair. We have some non-canonical types like type 3 and type 0, which are less frequent. And so this is the most frequent example of base triple, but actually we have a variety of different base triples, so like networks of 2 base pairs and 3 bases forming hydrogen bonds reciprocally with each other. So yeah, so base triple is also a 3D motif, but less strictly defined, and A minor is the most frequent type of base triple. Now, then you probably heard of G quadruplexes. So G quadruplex is a motif when we have in a single plane. So that is not like a double helix, but like a 4 base pair, 4 strand structure. So in each plane, so this is a 3D depiction. So in each plane we have 4 guanosines forming Watson-Crick-Hookston types of base pairs with each other. So Watson-Crick edge, Hookston edge, Watson-Crick edge, Hookston edge, and so on. And normally we have potassium ions in the middle of the 2 planes. So in between the 2 planes we have 1 potassium ion. The topology of the backbone may differ, and we have plenty of G quadruplexes in DNA, but RNA also forms G quadruplexes. And kind of the opposite motif, because it's often formed, so this I motif is formed by cytosines, and it's often formed on the opposite strand of a DNA. So here we have a G-rich strand, so the opposite strand is C-rich. So that's how these 2 motifs are related. So this is I motif, and it also has like 4 strands, but 4 strands of cytosines, but it doesn't form a single like tetrads in a single plane. Instead, it forms like alternating pattern of pyrimidine base pairs of CC base pairs. So we have C base pairs of a couple of strands, and here we have in between CC base pairs of another couple of the strands. So this is how it looks like in 3D. And I motif is DNA specific. So in RNA, it's very unfavorable. So we have among experimentally determined structures, we have just one known RNA version of I motif, but it was determined in specific experimental conditions to be formed. So in biologically relevant conditions, RNA does not form I motifs, but G quadruplexes are formed in both RNA and DNA. Now, some more. This is a king turn. Another also like very frequent and well described motif. The king turn is a couple of stems and normally internal loop between them. So here like one stem in yellow, two canonical base pairs. A second stem is in gray and green. So it has normally stacked non-canonical base pairs, most often AG hooks in sugar, up to 3. And then it continues with canonical base pair. So here we have just one canonical base pair. So, and these two stems form like a turn in the helical axis. So they're not coaxially stacked, but the direction of stems turns. And one of the strands is like a bulge strand. So it's a longer strand here, and it forms a sharp kink in the backbone. So that's why the naming. So we have a kink and we have a stem helical axis turn. That's why we have kink turn. So here the canonical interactions are these non-canonical base pairs of A and G, and this is actually, so this non-canonical Watson-Crick-Sugar AG base pair is a part of an A minor motif. So these two yellow residues and green adenosine form an A minor. So this is an A minor. And sometimes this nucleotide is also base paired with something else, and then this adenosine forms with them another type of A minor as well. Moving on, this is ribose zipper. This is like continuous sequence of ribose and base ribose hydrogen bonds. So it's like a zipper on your jacket, but it's formed by hydrogen bonds involving riboses. So ribose oxygens. So these are two. Here we have two hydrogen bonds, like one base and ribose interacting with one ribose, then the opposite, and it can continue, but we like normally focus on this like 4 residue representations of this motif, ribose zipper. This is an example of less well described type of group of motifs. So we have like loop motifs, which correspond to a single loop in a secondary structure, like a generator loop is a hairpin, kink turn is an internal loop, and on the opposite side we have so-called long-range motifs, which are formed not by a single secondary structure element or single loop, but by several of them. So this is an example of long-range interaction formed between D loop and T loop in a transfer RNA, in tRNA. We have instances of this motif in ribosomal RNA as well, in some other cases, but the canonical instance, which is the source of the naming, is the D loop and T loop pair of transfer RNA. So here we have two strands, which are two hairpins, and we have two long-range base pairs between the hairpins. So this is normally the canonical base pair in the CG. Here we have normally a non-canonical base pair. In this case, it's UG. And here we have a so-called like long-range stacking or a sandwich or an intercalation. We have different terms for this. But the thing is that this residue of one hairpin is stacked in between two consecutive residues of another hairpin. And these are like the characteristic interactions of the D loop T loop interaction motif, which is a long range motif. So as you already have an impression of, RNA structure is organized hierarchically, having like a modular architecture. So the entire 3D structure can be decomposed by secondary structure, so into a set of stems and loops. Stems, as we already know, are composed of base pairs, but in turn, like loops and in general tertiary 3D motifs of RNA also can be decomposed in the like smaller building blocks, smaller interactions. As an example here, a kink turn actually includes two stems and a loop. It includes A minors, at least one A minor, And in turn, A minor includes a base pair and a third base, right? So, and all this you can continue. So, like smaller interactions form small motifs, then small motifs form larger motifs, and so on and so forth until we get the entire 3D structure. So this is what I call modular architecture. Not only I, like many people call it modular architecture. This is a huge pain in the neck. So like this is the reason why the structural context is very important. Because once you like change the structural context, then you change the modules. It may result in dramatic consequences. This is a diagram from a paper I like, which actually is much more comprehensive overview of this concept of modular architecture. Here are like the network of different submotifs and how they form larger motifs. Like here, for example, we have U-turn motif, and here we have a G-array submotif, and they together form a G-array tetraloop motif. Here we have A minors, and this is a larger motif like G-array 11 and T tetraloop receptor interaction motif. So this is a motif where G-array tetraloop interacts with the tetraloop receptor, which is often an internal loop or just a helical fragment. And these are all the submotifs of this motif. So it includes A minors, it includes G-array tetraloop, it includes NT-platform, type 1 UA handle. What else? What do I miss? 1, 2, 3, 4, and a U-turn, of course, because it's a G-array tetraloop. And some more examples here. And on top, we have like really large arrangements of RNA structure. So I really like this diagram. It gives you like an understanding how different motifs compose larger motifs. Now the format. So the 3D structure is stored in one of the two formats. Normally we have a legacy PDB format, which is a very old format, but still like many tools, annotation tools or prediction tools operate on PDB format. Still like secondary 3D structure prediction experiments have templates in PDB format. So the PDB format looks like this. So it has different sections. The main sections with atom coordinates is atom section. This is how it looks like. This is an example of of a protein stored in this format. This atom section has this set of columns. So we have record name atom. We have integer, which is the number of an atom, then an element of an atom, then alternative location. So for each atom we have like several alternative locations where it is located. If we cannot determine it 100% to a single location, we can sometimes have like 2 or 3 alternatives. Then the name of the residue. So here it's arginine amino acid, identifier of a chain, number of the residue in a chain. Yep. 3 coordinates, x, y, z. Occupancy, so the percentage of time this atom is located in this position. So here it's all the atoms have 2 alternative locations with 50-50%. Then some B factor or temperature factor, which is like how well we believe in this atom, depending on some parameters for specific for X-ray crystallography. But now we use it for other experiments as well, but then less informative. And this is so this is not an element. This is atom name actually, and this is an element. So different carbons of a single arginine can have different naming. So this is alpha carbon, beta carbon, gamma carbon, and so on. And here the main thing, a very important thing, is that the columns cannot be shifted to each other. So we have specific indices for specific columns where they should be located, and they actually should be located there. And we have specific rules for text adjustment. Like for example, this atom name field has 4 positions, and if the atom name is shorter than 4 positions, it should be left adjusted with some exceptions. So it's really like very well strictly defined and very well documented, strictly defined format. And thanks to this specification, we can have no separators between like consecutive fields. So here, for example, alternative location and residue name merged together, but thanks to this specification, we can understand where is what. But yeah, so originally it was limited to 5 megabytes per file. Then this limit was not respected. And since the year 2014, the main format of the PDB databank, the main database of 3D structures, is the MMC format, which stands for macromolecular crystallography information format. This is a link to the specification. And this is a list of tables. It's also like a text format as PDB is, and this is a list of tables of different sections again, and we have like a counterpart of an atom section from PDB, where we first have a list of columns, and then we have here at least separated with at least one space all the values for a given atom. Here, like a single value of a single column cannot be empty, cannot be a space, because it will shift all the other values. So that's why for empty values we have these question marks. And the values can be separated by an arbitrary number of space or tabulation separators. Right. So visualizations. As I know it, we have two main. So like we have like Pepsi and Coca-Cola, we have two main visualization tools, standalone tools, IMol or Chimera X. And this is literally like half-half. Some people prefer. So normally it's like the first tool you choose, then you stick to it because you already know all the commands, you remember all the commands, how it works, and you don't want to learn the new tool from scratch. So it's one or another, like matter of taste. With web-based solutions, we have, I believe, more than two options, but I think these two are the most popular ones. So JSMol is a JavaScript implementation of JMol, standalone visualization tool. JMol was implemented in Java, so JSMol is a re-implementation in JavaScript, or actually like a HTML5. So like I'm not an expert in web development, so it works with it has to do something with HTML5 and JavaScript. And Molstar is more recent visualization tool. So currently Molstar is a visualization tool of PDB databank. It looks like this. Also, I think Molstar is more frequently used than JSMol. JSMol is older. So yeah. Databases. So we have a number of databases for motifs, as we already covered when we were talking about databases for loops of secondary structure. Because for loop motifs we have many databases and we have less number of databases for long-range motifs. So one example here for loop motifs already previously described, Motif Atlas from RNA 3D Hub. This is an example of a hairpin loop classes, one class, two class, second class, this visualization. This is Carnival, is an example of rare example of a database of long-range motifs. This is the link. Here it's based on the annotation of connected components in a graph where Fred annotated base pairs serve as edges between bases. So like in these diagrams, for example, we have two non-canonical base pairs, one canonical base pair. These are covalent bonds of a backbone, and this is like a recurrent connected component in such graphs, which are called long-range motifs because they include different elements of a secondary structure. So I believe this nucleotide comes from another element and these four come from a single element. Databases. So as I already mentioned, like Protein Databank, PDB is the main source of 3D experimentally determined 3D structures. It's called protein because it all started with protein structures, but it includes much less, but a notable amount of nucleic acid structures and nucleic acid protein complexes as well, DNA, RNA, RNA, DNA, RNA protein, DNA protein, RNA, DNA protein, and of course the majority of structures are just protein structures. So NAKB database, which is like, so previously we had NDB database, so now we have a new like re-implementation which is called NAKB. like roughly it has the same structures as PDB does, but it has an interface devoted to nucleic acids, so it has like base pair annotations, some like conformers of nucleotides, and normally PDB doesn't have any reasonable annotations of nucleic acids in it. And the main problem with these databases is that the content is highly redundant. So we have a lot of different ribosome structures, we have a lot of transfer RNAs, and we have not that many unique structures of RNA in the PDB. And to reduce this redundancy, to resolve this redundancy, we have some versions of like non-redundant subsets of PDB, of RNA structures from PDB. One most popular example is representative set from BGSU, which is a part of RNA-30 hub again. It has a single representative for a given molecule type of a given organism. So it have only one representative for like lysine transfer RNA of humans, and one representative RNA which is lysine transfer RNA of E. coli bacteria, for example. So that's why it's not a non-redundant. So previously it was named a non-redundant list. So the naming actually has it still in our list, but then it was renamed into representative sets because actually lysine transfer RNAs are pretty much conserved from organism to organism. So that's why it's rather representative than truly non-redundant, but still does its job well. Datasets. So when you work in prediction of 3D structures, a good benchmark datasets either custom subsets of PDB or sets of from this like prediction competitions. So we have we had for many years only RNA puzzles prediction competition, and since year 2022, since CASP15, CASP structure prediction experiment has RNA targets. So previously, since 90s, it included only proteins. Now it has RNAs as well. And like with times, with years, it stored a lot of targets and a lot of computational models, and you can just take data sets of this previous year competitions from previous years and take like the native structures of a target, predicted models of a target, and compare your custom solution with the solutions that participate in the predictions. So current CASP17 is still ongoing in the moment when I'm recording this lecture. So it's a competition from 2026, so the most recent one. Yeah, so these are not that huge data sets. So with RNA puzzles, so the round 5 included targets, I believe, up to 40. So we have just 40 targets in RNA puzzles throughout all the years. So we have not published ones, I think some more, so maybe now it's like around puzzle 70, but the latest ones are not yet described and published or analyzed. The CASP15 included 12 RNA targets, CASP16 included around 60 RNA targets, and CASP17 included more than 60 actually, but many of those this year was just for server prediction competition and not for human expert prediction. That's which is not that important from a dataset perspective. Now, and moving to 4D. So that was more or less everything on 3D, and of course 3D structures of RNA may interact with some other molecules. And I will just briefly mention types of different molecules RNA can interact with, and it's basically like all the other like biological molecules. So first of all, of course, metal ions. Metal ions are very important for RNA folding, especially magnesium ions. So many structured biologically relevant RNAs cannot fold properly without magnesium. So magnesium is an important environment for the proper folding. This is a list of motifs, motifs of how magnesium ions, hydrated magnesium ions, with this shell of 6 oxygens, 6 water molecules, interact with RNA. So we have some like named motifs of interactions, and so it's important that like positively charged metal ions should balance the negative charge, overall negative charge of RNA backbone. So without metal ions, positive metal ions, RNA would not fold properly because its backbone is negatively charged. So the source and the concentration of metal ions can direct folding. So this is an example like of how different concentrations of magnesium and potassium ions result in different formation of this sequence, of different folding of this RNA sequence. So with more potassium, we have G quadruplex formation. With more magnesium, we have hairpin formation. And this is another picture showing that potassium actually is important for formation of G quadruplexes. So it should include one ion in between two G quadruplex planes. So a single plane is normally called a G tetrad. The entire motif is called G quadruplex. So in between two G tetrads, we have one potassium ion. Then, of course, RNA interacts with small molecules of different kinds. This is an example I like, where we have a GNRA-like formation, but here instead of normal adenosine, we have an isolated nucleotide, isolated adenosine monophosphate, adenine nucleotide, and it intercalates between two consecutive purines of this RNA, and together they form a GNRA-like motif, but it's actually like an RNA. Formally, it's an RNA ligand interaction because this is a small molecule independent from the, not covalently connected to the RNA. This is another example how ligand changes the formation. So this is RNA without any ligand. This is one example of a structure where we have ligand bound to the RNA, and in some different conditions, this ligand instead glue four RNA instances together, so it can interact with RNA in different variants. And now it's a very hot topic in RNA therapeutics to identify small molecules, small molecule drugs that would target RNA structures. It's not an easy job to do. Then, of course, very biologically important and very rich in its variants are RNA protein interactions. This is just to give you a hint. This is a ribosome, and everything that is not yellow. So these yellow things are RNA. This and this and this, and all the other colors are proteins. So it has a lot of RNA protein interactions. Here we don't have any commonly accepted classifications of motifs or interactions or anything. At best, we annotate hydrogen bonds, and some people try to classify this is a work where people classified so-called pseudopairs, where kind of in the same manner as base pairs and non-canonical base pairs, people cataloged nucleotide amino acid base pairs, called them pseudo base pairs, which include two hydrogen bonds in each case. Of this yellow are nucleotides and green amino acids. But it does not go much more than that. So we don't understand a lot about RNA protein interactions, unfortunately. This is a snippet of SNAP tool annotation. So SNAP is a tool which is now a part of DSSR, and it annotates RNA protein interactions. And this is just an example, like this C arginine interaction, some parameters of just an example. So here people annotate some interactions, but we don't have even a single catalog of RNA protein motifs, which will catalog the most recurrent types of motifs. So this is really a wild west, and a lot to be done here. RNA, of course, a single chain of RNA can interact with other chains of RNA. This is a example of a T-box ribose switch interacting with a transfer RNA. So here we have two sets of interactions. Here interaction is derived mostly through stacking. So here we have stacking of two base pairs. Here are actually intermolecular base pairs. And recently we have an advent of papers which reported this so-called RNA nanocages, where this multimers of formed by many instances of single RNA chain. So this is I think gold RNA multimer, from a recent paper or rule, maybe, I don't remember which specifically. And this is like all the intermolecular interactions. And here in color we have one monomer of an RNA, so in rainbow coloring, and all the gray are different chains. So we have decamers, dodecamers, and larger variations. So a multimer can include like 10, 12, 14, and sometimes more, sometimes less. These are all the intermolecular interactions, which again mostly either canonical base pairs forming pseudoknots, or staking or non-canonical base pairs. Yep. So there is a debate of how biologically relevant these nanocages are. So maybe they just artifacts of experiment conditions, but maybe not. So RNA can form multimers, can form homomultimers, can form heteromultimers. Homomultimers mean that each monomer is the same monomer as here. Heteromultimers, like here, where the interacting chains are different from each other. So yeah, RNA, actually thanks to its modular architecture, the entire complex being a single chain is not that important. So actually we can cut some chain into pieces, and they will still, like the pieces will still form the same structure. And in terms of RNA DNA, important terms to mention are the nucleic acid hybrids, where we have helical fragments formed with one RNA chain and one DNA chain. Of course, normally like DNA helix is a B-form geometry, RNA helix form a A-form geometry, hybrids can form different geometries. This is an example of A-form. And apart from hybrid duplexes, we have so-called triple helices. This is how it looks like in 3D. So here, here we have a snippet of a triple helix. So in each section, in each base pair layer, we have 3 strands and one canonical base pair and the third nucleotide. And when it's like formed on a genome level, such triplexes of a single RNA chain interacting with a DNA duplex is called R-loop, and many like long non-coding RNAs form these R-loops with genomic DNA for some biologically relevant reasons. So just remember that we can have duplex hybrids and we have triplexes, and this is the last slide of the 3rd lecture of this course. See you at the 4th lecture.